might be. Thank you very much. Yeah, so a brief history of plant breeding at this point. And of course, we have been doing this since quite a while. About 10,000 BC, we already started in a phase called the Neolithic Revolution, where we transitioned from hunter-gatherers to farmers to do agriculture, basically. We got our head around how to sow seeds and how to breed plants in a way that we produced more food than we actually needed in a small group of communities that we were back then. And by that, we enabled people to do or to embark on different kind of labor, like, for example, where before everyone was involved in getting food, then after this time, everyone could also be a physician or an artist or become whatever they want. And it was maybe not over exaggerated to say that civilization as such, or culture as we know it, is in big parts only possible because we domesticated plants back then. But still, if we are honest with ourselves, there are also problems that came along with that, because as you know, we selected plants and we evolved them in a way that we wanted them to be, like in a way that mankind can use them more efficiently. And for example, the grasses back then are now big corns or maize plants that have nothing to do with a natural or wild relative. It's quite similar and comparable to what we did to the wild animals, which we domesticated and made pets out of them. So you all know that pets, as well as crop plants, have some similar problems like immune deficiencies, or the pug cannot really breathe properly, has hip problems. And it's really comparable to what we see in our crops nowadays. They are incapable of living without human help. So we need chemicals, we need fertilizer, and our caring on the fields for them to grow properly. And at this point we are right now, so we know what we did good and what we did wrong in plant breeding, and we have to decide where we are heading next. And in terms for us to understand NGTs, which is one of many plant breeding methods, we might want to start at the beginning where crossing was invented. So the most basic form of plant breeding, which was actually systematically analyzed by a monk here in Germany called Gregor Mendel. He set up the rules of inheritance for plants, which we still use in plant breeding today. And basically, what is done there is depicted here. You can, for example, we start off with a cultured plant, a crop plant, which has some kind of problem. For example, these brownish leaves might stand for a disease. And we want to cross this plant with a wild relative that has a disease resistance naturally occurring. And in the lower part, you can see the traits or the genes as small dots, and the red dot here represents the resistance gene that we want to confer. So we cross these two plants, and as we all have learned, when we cross mother and father, we can get a wild mixture, 50-50 of mother and father, and we of course get the resistance in the next generation, but we also get a lot of traits that we did not want, which can, for example, be a small growth, a reduced fruit size, or a bitter taste, or whatever. So what plant breeders do is, after this first crossing step, they back cross this new variety with the mother plant, and they do multiple back crossings until they reach a step where they say, okay, now this is a new variety we can bring to the market, which has this new resistance, but also a very low amount, or maybe some leftover traits that can come from the wild plant that we crossed in there. And you can imagine that these back crossings take very long. So it depends on the culture, of course. With tomato, it takes longer than, for example, with wheat. And we can breed new varieties in 15 or even more years, because it takes so long for breeders to do these back crossings. So you can imagine that using only the crossing techniques, breeders have to foresee the future, what will be necessary in a few decades from now for the fields out there. So in the 1950s, a new technique appeared, which is the mutagenesis. And here in brackets, you can see the word random, which will be more important when we compare the random mutagenesis to the targeted one later. Random mutagenesis basically means that we cause mutations. So we induce mutations in the genome, for example, of grapefruit. We did that. So there are over 3,000 commercially available fruits and veggies out there that have been randomly mutagenized and have been bred with using these techniques, and they are not labeled as GMOs, which is also a very interesting political or a legislative thing that we can stress later. But basically, mutagenesis works like this. You can use radioactivity or mutagenic chemicals or even UV light to induce mutations in plants. And you can imagine that in the next generation, some of these plants look even worse than before, because we absolutely randomly shoot against the genes in the plants. But of course, with some probability, genes are activated or are deactivated that lead to a new favorable trait in this plant. And for these, people select. So again, we get a new variety by back crossings here, which has a resistance, for example, caused by random mutagenesis, and then only a few other mutations remaining, or even they are not found at all, because they at some point the plants reach the market and have sometimes even hundreds of mutations left that have no direct effect on plant growth. But with this technique, and because of the back crossings, we also need 10, 15, or even more years to reach new varieties. Now, in the 1980s or 90s, there was the first time that we evolved, or we came up with a new breeding technique, which is distinguishable from these random ones, because it is targeted. And we know this as transgenesis, or as classical genetic engineering, where we use foreign genes or genes that are from distant relatives or even other species to transfer them to plants. And a prominent example might be the Monsanto 810 corn, which is resistant to the corn borer. The technique is like this. We have a crop plant here with a disease again, for example, and we take genes from other organisms, which might be bacteria, other distantly related plants, or whatever organism you want, and we can shuttle these genes into an agrobacterium, which is a soil bacterium, which also in nature is able to transfer DNA or genetic information into plant cells. We use these capabilities in the lab to shuttle genes into plant cells and to thereby get new varieties with these kind of chimeric transgenic DNA information, which can lead to, for example, disease resistance. And you can imagine, because this is not so random, we don't have to wait for a random mutation to occur. This takes way shorter to reach our goals, and we can create new varieties within only a few years. But of course, this with good reason is discussed at the moment, if this is a GMO or not, because of course this is something that in nature would not happen naturally, so to say. And from this transgenesis, we will now come to the genome editing, which is also a different technique, because genome editing also takes the genetic information that is there in the plant and doesn't necessarily depend on using foreign genes. And this is maybe the big difference to the classical genetic engineering that we had so far. There was, for example, in the EU, a Swedish researcher group who bred a potato, a low amylose potato with a favorable starch composition using CRISPR. And just to quickly explain how this technique works, we have in these crop plants, for example, inactive genes. As I showed you in the beginning, plants were, in the beginning of when we started to breed them, able to grow outside there and defend themselves. So it's reasonable to assume that they have genes inside them to defend themselves, but they kind of forgot how to do so during the breeding process. So with genome editing, we can search for these genes, we can cut at a specific site, and then make the plant have the plant repair these genes in a way that in the end we have a crop plant with an active resistance gene. And this goes without any foreign DNA. So we can breed plants from scratch, so to say, in order to get them in this case, a disease resistance or climate resilience, whatever the goal might be. And with this technique, we can reach new varieties or bring new varieties to the market very quickly as well. Now, maybe because we talk about NGTs today, we have to get some vocabulary right that also in media sometimes is switched or even wrongly cited. So genome editing is a term that holds true for all these techniques that we will talk about today, like CRISPR or TALENs or whatever they are called, while targeted mutagenesis is the thing that we are talking about in the EU proposal. So these techniques within the genome editing workbox or toolbox that allow for single base mutations, for example, without any foreign DNA. And I always compare this to a text program. So if you have the genetic information here in a text program, some of you might work with Microsoft Word program, might know this guy, and it's quite comparable to this guy because we can search for specific stretches in the vocabulary or in the words of the DNA by pressing Control F, and then we can at the specific site insert new information. For example, these red new letters, and this insertion of a new gene would be transgenesis, or would be even genome editing if we use CRISPR to find this place. But when we are discussing NGT1 plants, the ones that are in discussion right now to be deregulated in the European Union, these are not meant. We are talking about single base pair exchanges, which are very nature-like, and they are even referred to in the proposal as conventional-like. So the NGT1 plants, in comparison to the other NGT plants, like category NGT2, are really minor changes that could also occur like this in nature and are therefore conventional-like. And how does this work? We have the tool here, which is, for example, in the CRISPR-Cas method, the Cas9 protein, which is bound to a guide RNA. And this guide RNA, just like in our text program, can find the target point, like a search engine, where we want to do the base pair exchange, and then it's edited or cut at this specific site. And speaking of vocabulary, maybe it's even from a communicator's perspective, like myself, it's even not favorable to have all these different kind of abbreviations and terms that are really highly complex and maybe over complex for people to understand this issue. Maybe we could just skip going into too much detail, because people, of course, lose the track, and they maybe are not very interested in reading a text with all these abbreviations, because talking about zinc fingers, TALENs, CRISPR is fine, but they all mean the same, especially when we talk about the EU legislation. They're all methods to achieve or to produce these NGT1 plants, which are conventional-like, and in a way that they are targeted mutagenized. So they have point mutations within the genome, which could occur in nature as well. And they are, of course, distinguishable from random mutagenesis, because, as I showed you before, these techniques using UV light or radioactivity are really random. And these two are both legit breeding methods, but are in the current state of policy making treated very differently. So in the GMO directive, it says in an annex, organisms obtained by methods of mutagenesis, which have been conventionally used in a number of applications and have a long safety record, are exempted from this strict GMO law. This is what the European Court of Justice ruled in 2018. And this is the point where maybe the scientific community stood up and said, this is really not really understandable. Why should we do a deregulation or do an exemption, or even should not call those organisms GMOs that are randomly mutagenized and have hundreds or maybe thousands of mutations that we don't know what they are causing, when we have on the other side the NGT1 plants, which have maybe 1 or 2 or 3, maybe 20 mutations, but targeted on sites that we know, and these are then strictly regulated. So this is the kind of the discrepancy that we are in the middle of at the moment. And just in the end, I wanted to highlight a few of the chances and the risks that maybe come along with the genome edited crops. So there have been studies that showed, or many studies showed, that CRISPR and other genome editing techniques can be used to kind of rebreed crops that we already have. For example, this wild tomato within 1 generation was altered in a way that it had multiple flowers, enhanced fruit size, bigger growth, and even a more favorable content of lycopene, which is a very healthy antioxidant, so plant ingredients that can also be enhanced within 1 generation, where in the past we would have bred 10 or 20 or 30 years to get all these traits within within this plant. And of course, there have been economic studies as well, especially for smallholder farms in I think this was basically, or was mainly in Asia, where it has been shown that by the application or the implementation of genetically modified plants, of course, pesticide use can be reduced, and farmer profit also went up because these farmers didn't have to buy pesticides for their crops. So there are studies indicating that the rapid adaptation of plants is a huge chance for ecological, but also economic sustainability, and even socially speaking, societally speaking, sustainability can also improve by implementing these techniques in a group or in areas where technology is not so much implemented as here in our European Union. But of course, if we talk about risks, we also sometimes see an effect that the opposite of what I just showed you can be true. If we look at the US, for example, the implementation of maize and soybeans genetically modified led to a net increase of pesticide use, because GT, which means glyphosate-tolerant maize and soybeans, have been sold, and they, alongside the products that they need to be cultivated with, have flooded the market. And with this, of course, we see that on the one hand, the pests are reduced, but then the ecological aspects, for example, the use of pesticides, herbicides here, has risen. So the net increase of pesticides, for example, due to the economic interests of big firms, is a thing and a problem that we have to address in future. And also policy making can make its share and contribute to this not being promoted. And I think in the EU proposal that is on the table right now, herbicide tolerance is specifically or explicitly taken out of what is being deregulated. So in the end, it's totally reasonable and totally conceivable why there is such a conflict between the public, also between the whole organic sector, which, correctly says there have been old GMOs which did not keep their promises. Why should the new ones be different? And the academic world, where people like myself, early career researchers, see the potential of the new techniques. And of course, there are different fields of conflict that are outside the biotechnological issues, like what is about the patents? What about the freedom of choice? These are things that we also have to discuss, of course, but I would just want to emphasize here that these have nothing to do with the technique itself and have to be discussed separately. Because I'm running out of time, I'm just quickly showing you my opinion on why I think that NGTs are overrated, but also underrated in a way. I say they're overrated because the limits are set by nature. What isn't there in the genome can't be summoned. So in the NGT1 plants, we can only modify what is already there. And the impact of a single mutation is zero compared to the way we use land, right? For example, if you have monocropping, then there is, you can imagine that the impact on the ecosystem there is way higher, or is more impacted by the fact that the land use change has been made for making a pitch of agriculture here than a single mutation can ever have. But also, it is important to stress that NGTs alone cannot ensure food security, defeat hunger, or even mitigate the crises that we are facing at the moment. Because we need all aspects. We need agroecology, we need digitalization, we need people to waste less food, and only in a concert with all these different aspects and with the old knowledge that we have together with new technology, NGTs can really unfold their true potential. But then, of course, they're underrated because the breeding speed is just very unforeseen, and we need the speed right now in times of climate change that is already here, and we need new varieties as quick as possible. The rebreeding of old varieties can even bring us diversity in our fields, which means that we can have a more colorful landscape, basically, if we make the right political crash barriers to do so. And the chances inherent in NGTs overall contribute to the reaching of UN Sustainable Development Goals, as they are, of course, one of many tools to reach to reduce, for example, the greenhouse gas emissions by agriculture. Okay, with that, I'm at the end of my talk, and I hope I didn't lose you on the way, and I would hand over to Amrit for the paperwork. Thank you.
Thank you, David, for setting the scene. So I am going to share my slides. So hello, everyone. I'm just going to move this over there. Yeah, so my name is Amrit Nanda. I have a background in plant molecular biology. I spent 10 years in academia, and 4 years ago, I decided that I wanted to connect what's happening in the lab with actually what's happening at the legislative level, and how these different things are regulated and what kind of impact scientific breakthroughs can have on society in general. So I'll talk a little bit about the opportunities, a lot of them David already touched on, but also the challenges that we see. So basically, I represent an organization called Plants for the Future. We are, so full disclaimer also, we are a multi-stakeholder platform. So basically, our organization is made up of members representing the plant sector. These include academia, so plant science organizations, universities, research institutes. Also the seed and breeding sector, so the companies that are developing and breeding new plant varieties, and also the farming community. So by bringing these different stakeholders together, we can really come to a common agreement on what do we think is beneficial for the agri-food systems that is of interest for all the players, considering also the rest of the value chain. So among our many missions is to advocate for science-based policymaking to really support the plant sector and enable the translation of research for the benefit of society. So basically, I'm going to try to bring in here a little more of the kind of on-the-ground perspective of what NGTs can achieve, and also how different players can benefit from that in different ways, and how the legislation can help or not. So I've just looked a little bit about the potential of NGTs for breeding and food production, the GMO directive itself, and why it didn't work, and why we actually need a specific legislation for NGTs. And then I'll touch on the legislative process that's ongoing now, and where we are and where we're going. So basically, and I hope this figure is big enough for you to see, it's important to always keep in mind that regardless of whatever method is used for breeding, all plants undergo extensive selection and field trials, because often we hear the word deregulation, and I see David used it as well, and that can sometimes be a little bit misleading, because it sometimes implies kind of a free-for-all, that, you know, as soon as it's out of the GMO directive, you can do whatever you want and there's no oversight. But basically, all plants, conventionally bred plants as well, all undergo a very lengthy process, not only through the breeding process, but also for registration. So if you look a little bit at my figure, and I hope it's big enough for you to see, on the left, there's mutagenesis, which is the classical mutagenesis, the random mutagenesis that David described. In the middle, the crossbreeding, so by crosses. So again, David gave a very nice basis for how that works. And then the NGTs. So regardless of what method is being used, once the plant with the intended characteristics is identified, there's basically a lot of backcrossing and screening, etc. And again, David also explained it very nicely. You have to go through a lot of plants, and there's a lot of things you need to kind of sort through when you use random mutagenesis, because you have so many mutations at the very beginning. When you're crossing, you still have to sort through a lot, but there's proportionally less. And because with NGTs, the methods you're using are so precise, you know exactly what you're doing and what you're targeting, you can actually reduce quite a lot the amount of sorting that you need to do. So then when you're doing your back crossing, so again, you have many generations if you're using random mutagenesis, around slightly less if you're using crossbreeding, but much, much less if you're using NGTs. So regardless of that, you still basically want to go through selection and actual field trials, because whatever you see in your lab, obviously, in your controlled conditions, you want to make sure that it works in the field. So again, you go through several generations where you are looking at the whole plant. So of course, you're changing one trait, but you're also looking at the whole plant and how it changes the plant. There might be some other, side effects of what you changed. And this is where you would notice that, and you can actually sort that out during the process. So the breeders go through several generations as well. And then they go through the variety testing and registration, which also takes a number of years. And then their variety is approved from the market. So often when we're discussing breeding and the shorter time of NGTs compared to others, it definitely makes things shorter. But if the approval time, the variety testing and so on also takes a very long time, then in the end you lose the benefit to a certain extent of using it because it's taking longer. So while they are more precise, so basically the benefit of that in addition to speed is actually very interesting, because you need to basically sort less. You also need to grow less plants. So you actually need less resources. Your cost, of course, and your time is reduced, but also the land use, because you're maybe growing, a few hundred plants instead of growing several thousands of plants. So your inputs, of course, fertilizer, whatever, plant protection products you might need, water, all of that is reduced. So the speed is definitely something that has been highlighted a lot, but actually the lower resources is not negligible, because this makes it more accessible to smaller breeders, but also to lower income countries. And we're seeing this now, and I'm sure you've seen in the news, in Africa, they're really starting to explore this. There's more and more researchers that are starting to learn to use these technologies, and we already see field trials. So it really has a huge potential, because it makes it basically cheaper and easier, more accessible to breed using NGTs. So you will eventually get a bigger diversity of breeders and of varieties on the market. And one thing that I always like to remind, so we currently have what we call elite varieties, commercial varieties. So we have a handful, well, maybe not a handful, but we have, for example, wheat. We have maybe 10, 20 really good varieties, and those are used across the world. Of course, there's others, but they're maybe less used. By having technology like this, where we can really tweak very precisely, we don't need to go through the whole, crossing, back crossing, etc. We can actually take the traits of the elite varieties and put them in regionally important varieties, cultural heritage varieties, and we can actually make them more interesting at a lower cost, because the market is quite small. So there's not necessarily an interest unless the research institutes or the governments fund this. So that's something to think about as well, because it's actually a huge possibility. So basically, something I also wanted to highlight related to the variety testing and registration. And that's also something that has been a little bit of a bottleneck until recently, although the legislation might change with the revision of the plant variety or the seed marketing directive. But basically, once a variety is kind of ready, for testing and registration, basically you have this variety testing centers that can basically grow your crops and look at how they perform. And to have a new variety approved, you basically need it to be an improvement from previous varieties or current commercial varieties. And currently, the traits that are looked at are mostly yield, disease resistance, nutrient use efficiency, so the kind of standards. So basically, this creates a goal mark for breeders that they need to make sure that this meets a certain threshold. So traits like nutrition, flavor, processability, shelf life, it's not that they're, selected against, but, they get diluted. In the end, they're not the priority, because in the end, when a farmer wants to buy seeds, he's going to buy the seeds that can ensure him a good yield and a good disease resistance if he's in an area where he has those challenges. So he's going to look at something that's going to give him, maximum value for his money. If it's very, more nutritious, great, but he won't choose nutrition over yield unless he has some kind of compensation for that. For example, if there's a specific value chain where consumers are willing to pay more for that. So it's always something that needs to be balanced between, finances versus, sustainability, etc. And what's really interesting about NGTs, and we see this with the first varieties that have come on the market, is that because we can do these very precise tweaks and we can do them at a fairly cheap price, the first varieties that have come on the market are actually aimed at, for example, nutrition. So you have the high oleic acid soybean. So that's a more a healthier oil. So that came on the market already a number of years ago. I'm sure you've also heard about the GABA-enriched tomato in Japan. So that's also, GABA is a compound that's supposed to reduce high blood pressure. You also have Pairwise, which is a new company that's coming out with a lot of new interesting products that are all aimed at, so they had, for example, this concept of conscious greens. So a lot of leafy vegetables that are less bitter, more nutritious, and really trying to increase the consumption of fresh fruit and vegetables. David mentioned the amylose-free starch. There's also, I'm sure you've heard about the low gluten wheat that's also being developed by researchers in Spain, low asparagine wheat, so that we have a lower accumulation of acrylamide in breads and so on that are carcinogenic. And also Striga-resistant sorghum, for example, in Africa. This is under the initiative of Striga Smart Sorghum for Africa, which has just started field trials. I think it was last year. So there's really a big variety of things. So of course NGTs are still being used to increase yield, to increase disease resistance and so on. But there's so much more that can be done with it in combination, so that we can really diversify the traits and get a lot more out of it and healthier food systems as well. So coming to the GMO directive, I think it's important to realize actually why we need a new legislation for NGTs. And it's basically that, unfortunately, although it was well meant, of course, but the GMO directive has failed us. And that is also part of the reason that we did not really benefit from GMOs, because it's completely unmanageable. So since the adoption of the legislation, which was in 2001, there hasn't been a single new variety of GM approved for cultivation in Europe, not a single one. So that's really, for me, eye-opening. So for example, the GM maize variety that David mentioned, that one was approved before the directive came into force. And it's actually, so normally GM varieties need to be reapproved every 10 years. And it's basically still pending reapproval. So the company is submitting every year more and more material to the European Food Safety Authorities. But they still keep asking for more and more. So they're not taking it off the market, but they're also not approving it again, which is very strange. So that's been in a stalemate for over 2 decades, which is also a bit odd, I would say, to least of it. But something also to keep in mind is that in 2015, there was a clause that was added that basically member states could decide to opt out of cultivating GMOs on their territory. And currently 19 out of 27 member states have opted out. So the market is very, very small as well. So there is no incentive for companies to develop GM for the European market. So we are partly not benefiting from it because we're not making it doable for a wide range of companies, and that's why we get only big companies that have access to markets outside of Europe that are bringing in GMOs. And this something also to keep in mind, the kind of discrepancy of our legislation is that in the past decades, we've approved almost 100 GMOs for import, mostly for feed. So we import massive tons and tons of GMO crops, but we're not allowed to grow them here. So it's kind of, it's a very challenging situation. And so basically the whole point of having specific legislation for NGTs is because the GMO directive doesn't work. And when it was first discovered and used in plants, that's almost 10 years ago already, the stakeholders, so the breeding sector, they really they saw the potential for it, but they wanted to know how the EU would regulate it. And this happened not only in the EU, but all over the world. So there was a lot of discussions, but there were no official positions taken. So eventually, another case brought this whole discussion to the European Court of Justice, as David already mentioned, and they determined that all types of mutagenesis are genetic modification, which is true legally speaking, because random mutagenesis is genetic modification, but it's exempted from the GMO directive. And because it had its supposed history of long safe use, but because targeted mutagenesis, for example, was considered something relatively new, it was considered that it didn't fall under this long record of safe use. And then therefore they said, well, if you want to regulate it differently, you need to change the legislation. So basically, this is what has been happening in the last years. The member states asked the Commission to basically make a study of whether the GMO directive was still fit for purpose for what they called NGT. So that's when they took up this kind of acronym. And if it wasn't fit for purpose, to come up with a new legislation. So I won't go through all of that, but it's been a very long process, but it's also been a very, concise. A lot of things were taken into account, both ethics, impacts, both on member state level, but also stakeholders, etc. And that's when we came up with, or when they published the legislative proposal last July. I won't go into the details because I'm sure you already know it, but basically there are two categories. One is conventional like, and one is more kind of in between conventional and GM. And I think most stakeholders are interested in the category one. So basically, what are we looking at in terms of the legislative proposal? Where are we? So the Commission, they put forward their legislative proposal for NGTs. So that's almost 6 months or more than 6 months ago. And then it's being discussed by the European Parliament and the European Council. So basically, they need to come to an agreement amongst themselves, between the member states in the Council and between the Parliament, in the Parliament. And once they've done that, then they go into negotiations that we call trilogues with each other and with the Commission. And then they need to agree. So they each come with their own text, and then they try to find a compromise text that everyone is happy with. And once that happens, then they go back again with that compromise text, and then they need to adopt it within the Council and within the Parliament. And only at that time is the legislation adopted. So currently, what's happened so far in the Parliament, for example, so the leading committee on this file was what we call the ENVI Committee on Environment, Health and Safety. They had the lead, and then the Committee on Agriculture could give an opinion restricted to the Category 1 NGTs. So the Committee on Agriculture reached an agreed text in December, and the Committee on Environment reached an agreement on the 24th of January, which allowed to then take that text to the plenary vote, which happened last week. And so we're very happy to see that it was a tight majority, to be honest, but we did get a majority in favor of the text with some amendments added on. And I just wanted to highlight here that this is really a milestone that was reached, because this means that we have reached the first reading. Because as you obviously know, we have the Parliament elections that are upcoming very soon. And if the file is not, the final text is not adopted before the elections, that means it's the next Parliament, once they come in again, that they need to take this up. If they hadn't adopted the text in the first reading by that time, it meant that there was a possibility that the next Parliament could say, well, we're going to start over because it didn't, they didn't discuss it enough. It wasn't advanced enough. So they could actually have thrown out everything that had been done in the last 6 months and then start from scratch. But the fact that we have reached this part means that at least, we're there and they can continue from there. And so some of the amendments, and I'm not going through all of them, obviously, for sake of time, but some of the main amendments that were adopted in that text is the ban on patents for NGT plants. There was an addition, a very late addition, about traceability requirements throughout the value chain all the way to the consumers. I highlight this a lot, and we can talk about it in the discussions, but this is really a big challenge because, of course, we want to enable freedom of choice, of course, to the consumers, but at the same time, it needs to be consistent, because why should they get information about one breeding technology and not others, especially if the end product is the same? And also because of the challenges that something like this creates, because that means that all the products that come from NGTs need to be segregated. So farmers need to have separate silos. They need to sell to separate value chains. Processors need to have completely separate processing facilities. And we know from GMO, because that's how it goes with GMO, that's part of the reasons as well that they don't want to use GMOs, because they do not want to have this whole segregation. The costs are enormous. Another amendment that was added on was the requirement that Category 1 need to meet certain sustainability criteria, and they can't contain any unsustainable traits, for example, the herbicide tolerance. Another amendment that was added that we are a little bit concerned about is the fact that provisions for monitoring, including monitoring plan, which is fine, but the possibility to withdraw an approval decision if risk to health or environment is demonstrated, which is fine, but also if there is new scientific data supporting this hypothesis. And this kind of opens the door to uncertainty, because if new varieties are introduced and then suddenly they need to be taken out, if there is a proven risk, of course, that makes sense. Ideally, that wouldn't be the case because things are checked throughout. But if, for example, there's any hypothesis through science, that opens the door for a lot of potential pseudoscience, as well as we do see in some organizations that are trying to counter the use of NGTs. So this is something that, at least from the industry part, is a bit concerning. So I'm also almost out of time, but I'm almost done. So just to give an update on the council. So basically now the parliament is waiting for the council to come to their agreement so that the trilogues can start. And the Spanish presidency, which was the second half of last year, they really worked very hard to try to reach that agreement, but they didn't manage it just about that. So now we're under the Belgian presidency for the first half of this year. And they're trying to facilitate the final push. There's a lot of pressure because of the elections upcoming to get into the trilogues as soon as possible. But it's still they still haven't reached an agreement. And the main sticking points, it's mostly the patents now, but previously it was also the opt-out clause, because basically category 1 and 2, according to the Commission proposal, the member states were not allowed to actually ban the cultivation, because we need to have an open, a single market in the EU, and we need to enable these different varieties being grown everywhere. So there were some countries that are not happy about that. Some countries as well, the countries that are quite critical, they want full traceability and labeling. And then the ban in organics, that was very split. Some countries support this and some countries don't. And we see the same, for example, in the farming community of organic farmers, it's split 50-50. Many organic farmers would like to use category 1 NGTs because they see it as being similar to random mutagenesis, while some do not want to use it. So it's really a 50-50, and it's unfortunate that this has been put in the legislation here, because it's really something that should be addressed in the organic legislation for them to discuss amongst themselves, and to also give them the opportunity to reopen the legislation at a later time if they want to. But having it in this legislation means that they're locked out pretty much forever. So that is still a little bit under discussion as well at the council. So what is next is that if the council reaches an agreement soon, then the trilogues can commence, and either through the trilogues they reach an agreement before the last plenary of the parliament before the elections, which is towards the end of April, then we might have it adopted by then. If they do not reach that, then the trilogues need to resume after the parliamentary elections. If they do not reach an agreement, then obviously they cannot move to the trilogues before the elections. The Belgian presidency will not be able to finalize it. And then it gets a little bit tricky, because the next presidency is Hungary, and they are not supportive of the file. So it's very unlikely that they will push it. And the same for Poland, which even with the new government, they're still undecided. So it's unlikely that they would also push for the trilogues. So we're actually potentially looking at a resumption of discussions in the second half of 2025 by Denmark, who is supportive. So it really delays the whole legislative process. And it's important to also realize that once this legislation is adopted, the Commission needs to start developing the implementation legislation. And it's not until this implementation legislation is adopted that actual products can start coming on the market here in Europe. So this process takes an extra 2 years. So if we reach an adoption, for example, this year, then potentially by 2026, we can start getting new varieties approved for the market in the EU. If we're pushing it back, for example, 2025, then we're looking at 2027. So all of these things need to be, thought of in the back of our minds, because it is a lot of the opposition says, well, things are moving too fast, but it's actually taking a very long time, and it's being very taking all the steps properly to really make sure that we're doing this right. So I think that's it for me, and sorry if I was a little bit long. Thank you.
Thank you, Amrit, and thank you, David, for the presentation. Now, I think Maria will be moderating for the little time we have left. Maybe we'll stay 5 to 10 minutes longer, just so we can have a couple more questions. Please, Maria, go ahead, and maybe we should see if we can prioritize questions from the floor, or we can go with the questions that we've received online already.
Okay, thank you very much. Thank you very much for your amazing presentations and for your time and also for the questions. Now let's start. The first question is from SP Global. What is the impact of NGTs on biodiversity and ecosystems, and how can it be ensured that NGTs do not spread to organic land? Who would like to go first? David, maybe you?
Yeah, maybe I can just start and Amrit can add later. So the impact on NGTs to biodiversity or ecology was the question. It's this basically comparable to that what we had before. I mentioned that the mutagenesis methods changed now; they're more precise than before. But we had mutagenized crops already in the field, and I also mentioned that the way we produce plants, the way we do agriculture, has a much greater impact compared to the mutations within the genome. And well, sorry, what was the second part of the question again?
How can it be ensured that NGTs do not spread to organic land?
Right. Yeah, not to not speak too long, we really have to closely look at what culture we are speaking. So there are a lot of crops that do not pollinate with wind; they're just self-pollinators, such as most of the grains we have. And if there is some kind of contamination, so to say, from one field to another, then it's most of the times it doesn't really have an effect, because of course most of the farmers do not use their own seeds to sow them again. So they sell the foods as they are, and they are eaten without being changed in their texture or taste or whatever. So the effects most of the time would not be conceivable. And then of course there are rare exemptions where we see that, for example, corn or rapeseed is being pollinated from one field to another. But of course, organic farmers, like conventional farmers, have the same problems when they want to keep their varieties, so to say, clean from other varieties, might they be NGT or however bred. So there is a directive, here Amrit mentioned the seed marketing directive, that we have to keep a 99+% of pure or purity within the varieties seeds, independent of the breeding technique. So this is really a minor problem, which we have to discuss whenever NGTs are being brought to the market.
Yeah, I think David really answered that very well, so I won't add much, but I can just say that it's basically the traits that define whether there's an impact on biodiversity or not the technology used for it. So NGTs will have the same impact as conventional, and it is really how you grow it and what farming practices you're using that has the impact.
Okay, thank you very much. And now the next question is from Biocom AG. How can legislation effectively enforce control when NGT foods are not distinguishable from non-NGT products?
Yeah, and this is actually the main reason that this whole legislation was put in place, because the GMO directive that requires the whole traceability and so on is not enforceable because you cannot distinguish. And by the fact that you cannot distinguish, why should you? Because you're discriminating like products just based on the method they were developed. And we don't do that in any other case for breeding. So it doesn't really make a lot of sense. So the whole idea behind it is to avoid the situation where you find yourself in a whole court case potentially of trying to prove whether this variety was conventionally bred or not. So it's really to treat the conventional like ones that are not distinguishable as conventional.
Mm-hmm. Thank you very much. Now, David, this is a question really for you from Food Navigator. How will gene-edited foods be communicated in the future?
Yeah, that's a tough one, of course, because plant science has its advantages, because you always can show nice images of fruit and everything. But then on the other hand, it can be really challenging when we talk about misinformation, for example, or the emotional aspects of food, for example. So I think we have to find a way to communicate without all these acronyms. So we had NGTs, GMOs, etc. And this all suggests or already creates a kind of artificial image in the heads of people as compared to the way we see agriculture most of times. It's very idealistic, idyllic, I wanted to say, so a very green landscape and happy animals. And maybe people just have to embark and have to get their head around how agriculture is actually working at the moment, currently, and then speak for themselves or think for themselves where we want to head when we want to really reach a sustainable agriculture. And I think this can be done by proper science communication in a language that everybody can understand.
Thank you very much. Now we have the question from European Seed Magazine. What is the impact of ban on patenting for NGT plants?
Yeah, so there's not much I can say about that because that's outside of my, my realm of expertise, and we don't really discuss patents within our organization because we have multiple stakeholders that have different views. So even within the industry, there are different views on whether the ban on patents is a good or bad thing. And for the farmers, most of them actually support the ban on patents. It's I think the only thing I can say is that it really needs to be considered as a whole, because limiting it to NGTs for me seems, it doesn't make much sense, because again, you might end up with two products that are identical. One is conventional and could in theory be patented, while the other one is NGT and could not. And that for me doesn't make much sense. So I think there needs to be a look at more general approach to things rather than limiting it to a specific technology.
And from Expression Group, we have a question. Do we know anything about NGT and human health?
Well, I can just maybe quote the European Food Safety Authorities that have, they've done extensive studies also as part of this whole legislative process. And they did say that NGT plants that are similar to conventional basically are as safe as conventional. So again, the technology itself does not present a risk. It's what you do with the technology, and you can do, good and bad things with any technology, whether it's conventional or not. So there is no interest, of course, to have things that are, dangerous for people. And again, you have the whole process of selection, the variety testing, registration, and so on. So any kind of negative effects are always taken out before anything reaches the market.
Great, thank you very much. Daniela, do we have time for a few more questions? Or
yeah, sure. As well, if somebody might want to take the floor and ask further questions, we can also prioritize people in the call. I can see that people in the call also have submitted some questions, so maybe they also want to see their question responded.
Okay, I'll now see. Okay, apologize. So any climate change adaptation strategy involved?
Definitely. While in the past we mainly had the breeding goal of making higher yields, today, of course, we are trying to breed plants that need less fertilizer, less water, that grow longer roots. For example, there are many research projects on root length, so that plants are able to reach the groundwater more easily. So these are things that are definitely being evolved at the moment.
Yeah, I can maybe add also in the legislation. So there was the amendment. So that was something that the Commission had put forward that was removed during a previous vote and added on again, is linking the category 1 NGT plants to some kind of sustainability trait. The Parliament tweaked it a little bit because initially yield by itself was considered a sustainability trait, but now they say it needs to be yield plus something more. So yield plus resistance, for example, or plus tolerance to drought, and so on. And also more generally, not only linked to NGTs, in the update of the Seed Marketing Directive, they foresee in the variety testing and registration that I mentioned to update the list of indicators so that it also promotes the development of more sustainable crops. So that we're not only focusing again on yield and these different things, but that it's also more broader, so that breeders can bring a bigger diversity of traits to the market.
Thank you very much. We have also a question. Where does alternative food production systems like solar foods fit into this?
Well, I'm not so much in this topic, but I think with solar food it's meant that we have, for example, algae or other photosynthetic organisms within fermenters. And what we are talking at the moment is plants out there in the environment, right? So the whole EU proposal is for plants, and it's for the category 1 NGT plants that ought to be released into the environment. And solar foods, for example, also work with genetic engineering. So we have, for example, genetically improved bacteria or algae, but those are in closed containers. So this is a totally different topic, but of course goes hand in hand.
Okay, Daniela, do we have time?
Let's do 2 questions more.
Okay. Just give me a moment to see.
Maria, you're muted. You're muted.
Apologize. Who decides on what are sustainable and unsustainable characteristics? As it can be easily argued that herbicide tolerance is a sustainable characteristic.
Well, that is about to be discussed, I guess. So there's only the amendment to the EU proposal saying that there has to be some kind of sustainability testing. And of course, there are measures of measuring not only ecological sustainability, but also economic and social one. But the proposal does not specify on that. So this is something we definitely have to put out, but also have to think of sanctions or whatever means to do what happens if people do not meet these criteria or do not hold their promises, so to say. So this is really a very open question that cannot be answered at this point.
Yeah, I can maybe add, ideally, the whatever sustainability criteria that is put there should be linked to the sustainability criteria in the seed marketing directive, because again, it doesn't make sense to treat NGT crops, they should meet the same criteria as all other crops now. And so with the update of the seed marketing directive, it would make sense to link the two so that we don't at some point end up with very different, concepts of what sustainability is. And of course, it should be open to delegated acts, meaning that the Commission can come in and update that as things progress, because things that we might consider sustainable now are maybe less sustainable in future if we completely change our farming practices, for example. So there needs to be a little more consistency between these two legislations, I think. And of course, the opportunity to open it up and adapt it as we learn more.
Okay, thank you very much. And we have a comment. We are losing 40 to 80 billion US dollars each year to weed infestation. Yeah. And also, there is one more question. In 2015, Argentina became the first country in the world to develop regulatory criteria established whether a crop obtained throughout NGTs is or not GMO. Can we learn anything from it?
Well, basically, Argentina was really the front runner. They were the first country to come up with a specific legislation. And there's a lot of interesting things that they did. And I do believe the Commission looked into that as well, because they are also one of our main trading partners. So obviously, we want some legislation that actually allows also the continue of trade without having a lot of red tape in between. So not only them, now there's a number of countries now that have updated their legislation. So it's always important to have the big picture in mind. How does EU regulate versus the rest of the world?
And we have one more question. What are the 3 main arguments you would come forward with when having a discussion with someone who is against GMOs?
Sorry, can you repeat the question? I had a technical issue here.
Okay. Okay, just give me a moment. What are the 3 main arguments you would come forward with when having a discussion with someone who is against GMOs?
We have always been treating or breeding plants and changing their genomes since thousands of years. We have other technologies that helped us to do better in sustainability-wise. For example, solar power in comparison to coal mining, which is actually natural because it's out there in the ground. But we need the unnatural things also to be more sustainable. And thirdly, let's broaden our arsenal of varieties that we have out there by using NGTs to make wild plants into crops.
Amrit, do you want to add anything?
No, I think David,
Okay,
placed it very well. Yes.
Okay, and one more question. Can NGT be used to deal with invasive species and with rewilding efforts? Okay.
No, I think that would take me too far. Of course, there are gene drives discussed as well in South America, for example, to use NGTs in that way, but this is not part of the EU proposal that we are talking today.
Okay, that's all. We are running out of time. Thank you very much for your time, amazing presentations, questions, and let's not forget those who will be watching the recorded webinar later. Thank you and have a nice day. Daniela, back to you.
Yes, again, thank you for staying over time. We will try to see if some questions will be responded as well, maybe via email together with while we will be sending the material. We have a couple of questions that were not responded, so maybe we will make the favor to the journalists as well that did not get the chance to get their question asked. Again, thank you and have a lovely day. Bye.
Thanks, Daniela.
Thank you too.